Please join me in welcoming Dr. Wu.
Thank you very much, Chairman, and also thank you very much for the committees, particularly Dr. Abichat, Chairman of the committee, to invite me to give me an opportunity. And it's my pleasure to talk some work now carrying out in our level. As everybody know here, understanding the nature variation in rice is of great benefits to its breeding program for improving yield quality and sustainability with available adaptation to diverse environments. And many thanks to the rice community who has supplied us completed and high-quality genome sequences from the Japanese rice, Nippon barley, and also many thanks to the rice community who has, collect a huge number of the rice varieties, land races. So here, more than 100,000 land races and varieties stocked in some bigger centers like Italy, China, and Japan. And also thanks many researchers in rice community. They so far has made a lot of genetic materials such as F2 mapping population and RA lines, PI lines, and also CS lines, and also so many things to the developed advantage of the NGS, the next generation sequences, technologies that has made us to have genome sequences and for such as GWAS analysis and as you know, we have had yesterday from the Chinese group, Binhan's group, and also from the BGI. However, no comprehensive studies on the associations between nature occurring nucleotide variations and phenotypic changes in rice still remains as a very big challenge. As you know, so far only several hundreds of genes have been cloned and whose function has been analyzed. So if we consider rice has more than 30,000 genes, so there's only maybe less than 10% of the total genes. So to carry out, to face this challenge, probably definitely we need to have integrated platform with effective systems, for example, to including to select materials, to do the genotyping and gene sequence and link to discover the sequence mutations and then link them together. And of course, and as a kind of, for example, as gene expressions and so on. I think yesterday, Dr. Liano has talked something about the phenotyping and the selection materials at this place on his plenary presentation. And today, our main focus on our current progress on the genome resequencing and the database construction and the development of some tools for research to use. Okay. So firstly, we aim to obtain information for natural sequence variation using a diverse panel of rice accessions, including those varieties have been used as donors for these line constructions through. And the method we use is, as I mentioned, we use NGS technologies and to sequence deeply multiple accessions of the genome. And second, we are now in build a database that contain fundamental information of natural sequence mutations of rice genome, and then we developed a browsing tool to display this mutation status, mutation informations, and also contents with a genome view. Everybody can see, can look, have a look at, and can know where and what kind of mutation occurred in the right genomes. And finally, it's a small topic, so just in order to support our, progress on the genome isolation and function analysis of the genes, we also contract back libraries, for analysis of chromosome structure changes. For example, in some regions, maybe big changes, so we cannot know the details, the structure sequence or structures of those regions when you use Japonica as a reference. So sometimes we need a bag to sequence, and also, of course, we need a bag and need the DNA fragment for complement test to know to check the function of the tagged genes. Okay, it's important to select the materials. And several years ago, in our institute, a set of RFLP markers were used to screen more than 3,000 of rice, landrace, and Fridays stocked in nearest resource centers. And from these more than 3,000 samples, we select based on the genotyping results, and we select more than 150 samples, which represent most of the diversity. And then we used more markers. For example, we designed new markers based on the properties of the P sign or reg transposon, insertion, deletion, polymorphisms. And we did the phylogenetic analysis again. So we got this kind of trees. And so you can see here, from these trees, of course, also we also did some phenotyping of these materials. So from these trees, we choose 32 rice Fridays and 2 white rice accessions. As here showed in yellow, that's 2 white rice. And also here you can see some circles in red color, which means Fridays, which has already been used for the contraction of CSR lines. Okay, here just show how where these landrace or Fridays coming from, which. Okay, so as I mentioned, we actually surveyed some traits from our samples. Here just show, for example, the heading date. You can see here, and there's the height of the plants, and here's the green length, and here's green widers, and so you can see the quite diversity among these varieties. Okay, next I would like to move to the sequencing result to describe our current results. And the method is very simple. I don't think I need to mention so much. So I will just collect the leaves and isolate the DNAs. And here all the samples were sent for the short NGC sequencing using NGC short type sequence, and only for 2 varieties, Caceres and Niva, like ours and this is indica, you also use long read type sequences, and for I think not so deep, 5 or 6 X of sequences, and then we use the data, we contract pipelines, and then analyze the database. And the next shows the current results of our genome sequence. Here is the names of rice Fridays, and here is the origin, and here's the temperature of molecular or tropical Japonica and some indica oils, aroma indica oils, only thing yes. And also there are two, exception for white rice. Anyway, it's too small maybe to say it doesn't matter. It's just how many nucleotides we got from individual accessions of varieties. Anyway, short sequence amount, of the short sequence at least 36 times of genome equivalent were obtained from all of the cultivars and wild rice ecosystem shown here. Okay, as mentioned, we need, because we got so huge amount of sequence data, and we need an effective, method to analyze the data. And firstly, we contract a pipeline, as shown here. Yeah, and low data, and then we do some trimming of sequences, and then we mapping to reference sequences, and then we got the alignment data and do some filtering. That means to remove all the multi-hit leads, and then we do local arrangement. And also removing pitch-up duplicators, and then at this stage, we use data and BM tool, BM file, and so firstly we can calculate the depth, how depths each regions will lead in each varieties or accessions, and also we can we do the variant calling, and then we got the variant data. And here, and from here to here, if you we just put the data and then we put one key part, okay, so we can get the results. This will automatically. Okay, and then it's not enough. We need to see detail of the mutations. So we developed a Genome viewer to display our results at a very more, detailed. Okay, that I will tell you later. Okay, here's the result. This slide shows how much, the genome was covered by the short read sequence. We mapping this result coming from the reference mapping here, and the red color, the blue color. You can see it means the coverage for the whole genome, and then you can see here, and the ranges from 81 to 94, and from here to here is just the varieties from the Japonica groups. So it's not surprising to know the coverage is very high because we use the Japonica, use the Nippon barley as a reference. But however, you will see here there's a indica or our varieties, so the coverage go down to about 80 or 85%, something like that. Different with whole genome, the coverage of the genetic region is very high, and the difference is not so bigger. Anyway, whole genome coverage in indica and all the varieties is lower than in indica varieties by about 10%. That means when you use Japonica Nippon barley as a reference, about 10% of the genome regions, so from indica or all varieties cannot be, analyzed. Okay, it just shows the mapping here using the BWA, and here you can see the molecular leader mapping to the regions. And here, you from the readers, and you can get a snippet information. Yeah, it just not so. And this slide summarizes how many SNPs and indices we have got from our receiving samples. Here is the red. And again, when because we use Nippon barley as a reference, so the SNPs or indices was fairly the rate was fairly low. Particularly for this varieties, because this variety as Japanese are growing in Japan. And I said this varieties I think is coming from East Asians. It's like a tropical Japanese. But here is a Japanese indica or our varieties. So the rate is very high, and a similar pattern you can see from the genetic regions. Okay, I want to repeat. So about 10% of the genomes from the indica was very difficult to analyze when you only use the Nippon barley as a reference. So now we try to construct show the molecular sequence from indica, and also here I just show a sample of our efforts on the construct such kind of molecule from castorous rice, as all varieties. We sequence using the long read type NGS 454 to I think 6x genome equivalence, and then we do the de novo assembly. And then do some sequence trimming, and then we got the contigs, and then we use the short read from the Illumina, the sequence data as I reported before, to mapping them to edit errors to within this contiguous. And again, after we get the contiguous, again we mapping the raw data from the Illumina again to this contiguous. Particularly in this case, unmapped remains, and so we did again use another assembler tools to do the assembly again. So we can get large sequence contiguous, and also because we have the back end sequence from CASAS, so we also use the kind of information. Finally, we control scan fold. And the next, this slide shows results. And here, the sequence assembly. And when we finish the scan fold, you can find the N50's length about within 12 kilobase. And the total sequence is now go up to 337 megabase. This means it's 32 megabase longer than that we obtained only from the reference mapping. Okay, next I think how we try to display our results, and this is particular. This is a window for sequence diagrams. You can see the colors, the different. The density of the color means if you have a dense black, this means high sequence depth, and if the thin colors, it means the low sequence depth, and one block I think is 1 KB. So, and also here, so we this window shows a snip frequency. Snip radius was 1 block 1 KB. For example, the dense blue I think is maybe 1 KB. So I think it's too small, 1 or 2 snippets within 1 KB region, something like that. Anyway, we divide to different, according to the rate, different levels of. It's a very high rate and it's a low rate. Okay, time is coming. And here's the insert side. You can read, here means here you can see our insert. And again, this is mutation. So because we have mutation, we look at into the contents of the mutations, which, for example, mutation whether would lead to the changes of amino acids or frame shifts or SNSM. So you can, through this window, you can see where and which kind of mutations occur. For example, in large this window, you can see here, yellow means level 3. Level 3 means synonymous substitutions. I'm sorry, yes, at the start, and snippets at the exon regions, and also some. And level 4 means the more damage, the mutations lead to more damage non-simultaneous encoding regions and similar. Sometimes level 5 that means the insertions and leading to the frame shift, and or maybe to leading to a production of a new stop premature stop codon, something like that. Anyway, 0 is a mutation occurring in the intragenic regions. Anyway, so the degree of damage of the mutations is gone up, from 0 to 5. You can see. Okay, so next 2 or 3 slides just show. I think Dr. Yano has already shown yesterday here. It is here just show 2 of slides. Here we use our data to, for example, 1 year ago we use 12 F2 populations to mapping QT heading data, QT of heading data. We found, for example, here you can find from different populations the name of the Fridays. So they show different effects for increase or decrease the data for heading. And okay, I'm sorry. So use our data, you can find this kind of effects are obviously caused by this kind of mutations, like stop because of the insertion, deletion, and this one only deletion. And this is a case for the major factors, and also we're now using this kind of work, and it's now ongoing. We use the CSS lines to map some genes heading with minor effects. For example, here you can find we have a lot of loci, and then we can use the data from sequences. This as showed here. For example, this kind of mutations means level 5 or 4 or 3. So we can use this data to help our gene isolation. Okay, finally, here's just a result of contraction back laboratories at our institute. Okay, sorry, the summary. Okay, genome from a total 32 characters and 2 wide rice accession were deep resequenced using short time array of NGS. Mapping of short read sequence revealed coverage of 81 from 8 to 94% for the whole genome, and 92 to 98% for the genetic regions against reference sequence. Numbers of about 2.8 million SNP sites and 0.4 million injured sites were detected on the genome-wide. A genome viewer was developed to display the frequency and the content of sequences mutations with the effects on gene structures. The no assembly of short reads and long read sequences generated by NGS from clusters led to 137 megabase genome sequences. And now we are continuing our work to link the sequence mutation to the phenotypes. And I hope next time I can report some new results. Okay, here just acknowledgement. These people did contributions to material preparation and gene sequence, and these people did a great contribution to the database analysis and the view, and it's a contractual genome view, and these people did work on genetic analysis and the back construction here. So, thank you very much.
Thank you. Although time's late, we could still entertain 1 or 2 good questions. Okay, if not, please join me.